Hi guys, welcome back to my channel. Let's talk about some protein purification lab technique, gel electrophoresis. So in today's video, I'll be talking about 3 gel electrophoresis, being the native one, the SDS page non-reducing one, and the page reducing one, okay? But just so you know, there's a 4th one called isoelectric focusing, which is not going to be included in today's video, okay? And then also, electrophoresis is not just applying to proteins. We can also separate DNA and RNAs given their different sizes using electrophoresis, right? But today's video will be focusing on proteins, okay? All right, now let's look at the setup. So we see there we have some power that supply the electric field, right? So that we have this constant electric field where the currents can run, okay? So the negative side is the cathode side, right? And then the positive side is the anode side, okay? All right, so the protein that you load into the sample wheels, they're going to travel this way from the cathode to the anode, from the negative side to the positive side, right? And then we usually think that if the protein is relatively small, it would travel faster. And then if they are relatively larger, then it would travel slower, okay? All right, so this is the basic idea. And then just to relate to physics really quickly. So if in a passage that they show you, okay, this power supply shows a voltage of 10 volts, okay? And then the distance between the positive and negative electrodes is really 0.1 meter, let's say. And then how do you calculate the electric field, right? So this constant electric field is really what? E equals V over D. So it will be 10 volts over 0.1 meter, okay? So that will be the answer, okay? Just a little bit connection to physics, okay? Perfect. Okay, so another thing you need to know about this setup is that it's really different than a galvanic cell, right? Because the galvanic cell, the cathode is the positive side, right? The anode is the negative side. But this is really acting like an electrolytic cell, right? Because the galvanic cell is really this spontaneous reaction, but this electrolytic cells, this setup, this gel electrophoresis, like an electrolytic cell, is really relying on the power supply, right? It is really a non-spontaneous reaction that's happening, so that it's flipped, okay? Perfect. All right, so this is the setup that you need to know. Now let's interpret the results, okay? So after the protein, after they migrate this way, right? And then when I see the results, I then put it under some x-ray, then I'll see that, okay, some bands are here and some are here, right? So if I say I have a 2 protein, this is protein A, protein B, this means that protein A is a 60 kilodalton and protein B is a 55, because this is my ladder, right? My ladder is a reference for me to tell how much does my protein weigh, okay? Awesome. So what is the special about native gel, okay, versus, the SDS page gel, okay? So the difference is, so the special thing about native gel is that it doesn't change the nature of the protein. It doesn't change the size of the protein, the charges of the protein. Whatever you transfer to the sample wheel, it would just let it migrate the way it is, okay? So let's do a couple examples. Let's say I have 3 proteins, protein A, protein B, and protein C, okay? So protein A weighs 60 kilodalton, and then protein B weighs 30 kilodalton. Protein C weighs 30 kilodalton as well, but really with more negative charges, okay? All right, so let's see how they migrate. So protein A, as we can imagine, it will migrate to here, right? The 60 band. And then the protein B will stop here at the 30 kilodalton ladder, okay? And then protein C will also stop at 30 since it weighs 30 kilodalton, right? But the difference is that since protein C carries more negative charges, then it's more attracted to the anode positive side, right? Again, the protein travels from the negative side to the positive side, from the cathode side to the anode side, so that protein C would travel faster than protein B due to more negative charges, but they will eventually end up at the same level, okay? Awesome. So this is my native gel. You can really just tell the differences of the protein by their different sizes, right? And you can tell their different charges, their charges by the speed of their migration, okay? All right, so now let's move on to the SDS page, okay? So SDS page, we have 2. One is a non-reducing, okay? Non-reducing. Another one is the reducing one, okay? So the non-reducing one has some special detergent, okay? So they are not like the native one. The special detergent can really break quite a few intermolecular forces. Like for example, high hydrogen bonds, right? And then what's the other one? The other one is some ionic attraction, right? Okay, and then also like the hydrophobic attraction, like the London force, okay? So these are the intermolecular forces the SDS page the detergent of SDS page can really break, okay? But this is the non-reducing one. But the reducing one, not only can it break the hydrogen bonds, ionic attraction, and London force, it can also break the disulfide bond. Okay, and then which protein, the disulfide bond looks like that, right? Which protein can have this disulfide bond? It's the cysteine, right? Okay, so as you can see from the picture here, so once again, these are the letters, right? These are the reference point. And then we have protein 1 and 2 and 3, 3 proteins here, right? So they're about like 130 kilodaltons, right? And then you see that when I run the reducing gel this time with the same proteins, that they're broken down into more fragments, meaning that there must be some disulfide bonds in this in all 3 proteins here, okay? All right, so that's the SDS page non-reducing and the SDS page reducing, okay? And then 2 other things you need to know is that neither SDS page non-reducing nor the SDS page reducing would break a peptide bond. So they never break peptide bond, okay? It's also a covalent bond like the disulfide bond, but it's never broken, okay? It's fairly stable. And then if it would be broken, then we lost the point of the experiment, right? Because the purpose of the experiment is really to purify protein, to get the protein of our interest. If the peptide bond is broken, then it's not a complete protein, right? Okay, so the second thing you need to know about these SDS pages is that they turn all the protein into negatively charged proteins, okay? We have some negatively charged, we have the net charge of the protein can be negative, can be positive, can even be neutral, right? But the special detergent would turn the protein, and then also the pH of these buffer, these gel will turn the protein all into negatively charged. So that meaning that from SDS page gel electrophoresis, you can really not tell the different charges of the proteins, but you can only like tell their size and weight differences, right? Whereas the native one really take both size and charge into consideration, okay? All right. So that's the basics, okay? Now let's talk about protein structures for a minute, okay? Before we dive into 3 very challenging MCAT questions, okay? All right, so the quaternary structure of the protein, we can have monomer, meaning that there's just 1 subunit, right? Or you can have a dimer, meaning there are 2 subunits, or trimer, 3 subunits, or up to tetramer, the hardest it can get on MCAT, okay? It doesn't go beyond that. So tetramer, 4 subunits, right? So the dimer, trimer, and tetramer, they can really also have like a homodimer, homotrimer, homotetramer, or heterodimer, heterotrimer, heterotetramer, right? Homo means the same. They're the same. Hetero means they're different. So let's do an example, okay? So let's draw it out. A tetramer with 2 homodimers. What does it look like? Okay, so I'm going to draw the first homodimer. The first homodimer, I know dimer, 2 things, and homo, so they're the same, okay? So that's 1 homodimer. Let me draw another homodimer. There you go. So I connect them together. But voila, it's a tetramer, right? 1, 2, 3, 4, okay? 4 subunits. Okay, now let's draw the tetramer with 2 heterodimers, okay? So let me first draw the first heterodimers. So dimer, 2, but hetero, they're different. So 1 and 2, okay? That's my first heterodimers. And then I'm going to draw the second heterodimers, okay? And then they connect together will make a tetramer with 2 heterodimers, okay? All right, I hope that's helpful. Okay, with the background knowledge, now we're going to dive into some problems that I made up, okay? And then I think the problem that I made up are more challenging than what you will see on MCAT, but it's always good to start from the hard, right? Okay, so let's read the problem. So I have a protein. It's a 2000 kilodalton tetramer, okay? And then I have 2 identical heterodimers, okay? So very quickly we know that, okay, heterodimers, one heterodimer, two heterodimer. It's a tetramer. Oh, this is my protein, and it weighs 200 kilodalton. But I don't know like how much does each subunit weigh yet, right? I just know like this guy, if this weighs X kilodalton, this would weigh X as well, right? If this is Y, weighs Y kilodalton, this other one would be the same because that's two identical heterodimer, okay? Perfect. All right. Okay, so this protein, I did two modifications to this protein. The first modification is at the number 110th amino acid, I replaced alanine with a cysteine, okay? The alanine is gone. I replaced it with cysteine, okay? And then the other modification is at the 112th amino acid of this polypeptide chain, I got rid of the cysteine and I replaced it with the alanine, okay? So the question is, I show you that I run all, the wild type and then the modification 1 and 2. I run 3 types of gel electrophoresis, okay? And then that's as a result as you see the picture here. And then the question is asking, which one of the following is true? A says variant 2 means modification 2, right? Contains disulfide bond. B says the variant that increased the transporting rate of L-carotene is stabilized by hydrogen bond, hydrophobic, and ionic interaction, okay? So this is really, I'm saying variant 1, okay? Modification 1. But I'm just coming up with this fancy sentence because this is just MCAT style, right? Because you have to find out from the passage that, oh, the modification 1, the variant 1 type is really the one that the passage tells me, or the graph or the table or the chart tells me that, oh, it shows that it increases whichever one that increased the transporting rate of L-carotene is the protein of my interest, okay, for answer choice B, okay? All right, so C says the only covalent bond of the wild type is its peptide bond, and D says the wild type contains disulfide bonds, okay? You can pause the video and take as long as you need to solve this, okay? This is really challenging, otherwise we're gonna dive into this, okay? So I'm gonna delete this for a minute, okay? I promise I will show this again to you, okay? Okay, so we are going to analyze this, okay? We're going to dry everything out, okay? So first, I know that my wild type, as the question tells me, I have this 2 identical heterodimers, okay? And then they weigh 200 kilodaltons, right? And then this checks, okay? This checks. Okay, now let's move on to the SDS non-reducing. SDS non-reducing. Now I see my wild type is broken into 2 pieces, right? And then one piece weighs 160. Another piece weighs 40. So that tells me what? That adds up because 40 plus 160 is 200, okay? So I would say, okay, so I must have this and then this, okay? So if this weighs 40, then this will be what? This will be 20, and this will be 20. And then 160 divided by 2, then this must be 80. This must be 80, right? So this really add up to what? 200, okay? So this is what the non-reducing tell me. From the wild type, I just know this, right? I just know they're all connected, right? Okay, but now I know they're broken into 2 parts. The 2 parts, one part is a 40 kilodalton, as I show here, right? Another part is 160 kilodalton, so as I show here, the other subunit, okay? All right, so this is a non-reducing. Okay, now let's turn our attention to the reducing one, okay? Okay, so when we turn our attention to the reducing one, you see what is going on. So now my protein, okay, really got broken down into even more, okay? Because you see, I have 2 fragments, okay? One is 80, one is 40. And then you can tell that 80 times 2 is 160. 20 times 2 is 40, right? This is really what the non-reducing one shows, okay? So what does that tell me? That tells me now they're really broken into 4 pieces for the reducing one, okay? It's really like that now. So I have this is 20, 20, 80, and 80, okay? And then I have 2 of this subunits, okay? And then 2 of this, okay? So this is telling me what? This is telling me something about the bond, right? So we can say that the non-reducing, really these bonds, this and this, because they're broken here, right? So these are whatever intermolecular force, right? Hydrogen bond, ionic, whatever it is, right? But then we know that these other two bonds, they are not either hydrogen or ionic bond or London force, right? They are what? They're disulfide bonds, because only the reducing gel electrophoresis broke those bonds, okay? All right, so that my conclusion is these two are the intermolecular force, okay? And then I'll say these two are the disulfide bonds, okay? All right, so this is my wild type, okay? Now, let's analyze the modification 1, okay? So the modification 1, okay, still the wild type still gives me this, right? It's a 200 kilodalton, okay? Nothing's broken because the native one, right? We're now doing the native one, okay? So these are all the native and native, okay? Now let's look at the non-reducing, okay? So what happens to non-reducing? Okay, so the non-reducing, it's still 200. So meaning that none of the bonds are broken, right? So non-reducing still gives me this. Still gives me this, right? Because if it's broken, I'll have more fragments. But no, I don't. It's still 200. So meaning what? This tells me what? This tells me all the bonds here, they're all disulfide bond. So that the non-reducing one cannot break, right? Does that make sense? Okay, so if this is the case, then we will have more fragments when we see the reducing, when we run the reducing gel electrophoresis, right? And then we see that this is the case. Now the 200 kilodalton protein is broken into really just like my native one, right? It's really broken into 4 pieces, right? One being 20, 20, 80, 80, right? So I have a 2 times 20 equals 40. And then this is a 2 times 80 equals 160, okay? All right. Now let's move on to the modification 2, okay? The variant 2. What's going on with variant 2? Variant 2, also we see that the native state is also the same thing, right? Okay, this is the native one. Okay, but what about the non-reducing one? So you see the non-reducing one here, it really just looks like these we've analyzed, right? Meaning what? Meaning that when we run the non-reducing one, already all the bonds are already broken. So this means what? This means the modification 2 really make all the bonds, they're all what? None of them are disulfide bonds. In other words, they're all those hydrogen or London force or ionic bonds, right? So none of them are disulfide. Okay, none of them are disulfide bonds, okay? So let's check if that is the case, okay? Okay, let's see the reducing one, okay? So, yeah, I mean the reducing one and the non-reducing one, they're really the same, okay? So really this protein, as we see here, it's already, broken into 4 pieces, okay? Let me tell the difference. It's already the 4 pieces, okay? And then the reducing one, of course, it breaks all the bonds the non-reducing can break. So I'll still have the 4 different subunits, okay? All right, so I hope that makes sense. So after analyze it this way, now I think we're ready to move on to the answer choices, okay? So again, the answer choice says, A says, variant 2 contains disulfide bond, okay? So variant 2 is the modification 2, right? We said it's a non-disulfide. So A is not true, okay? B says the variant, which is a variant 1, okay? So variant 1, what does it say? Oh, it's stabilized by hydrogen bond, hydrophobic and ionic interaction. Is that true? No, right? It's really stabilized that all the bonds are disulfide bond. So B is wrong, okay? And then C said the only covalent bond of the wild type is its peptide bond, right? Is that a case? No, right? For the wild type, we have a peptide bond, but that is the covalent bond, but we also have disulfide bonds that is also covalent bond, right? So C is wrong. And then D says the wild type contains disulfide bonds. Okay, that is correct. Okay, so D is my best answer. Okay, perfect. So when I designed this problem, you can really see my my logic here. See the modification one of the first modification that I turned all the alanine to cysteine. Basically, I'm saying, hey, I'm supporting the disulfide bond formation, right? So that's why the modification one, you have all disulfide bond, right? And then the modification two I did, I got rid of all the cysteines, okay? I replaced cysteine with the alanine so that basically I'm saying that, oh, I don't want that many disulfide bonds to be formed. So you see that from our data analysis, this modification two, variant two, is really having no disulfide bonds, okay? All right, okay. That's a lot, okay? If you got the answer correct, kudos to you. That's really rough. Okay, now let's do something mellow, okay? Okay, let's take a breath, and then this is really easy, okay? I promise, compared to the other one, but it tests you different things, okay? So the question asks, how many amino acids, I'm missing it, how many amino acids does the smallest subunit of protein a tetramer have? Okay, all right, so I know that my protein X, it is a trimer, right? So three things. I don't know what kind of bond, okay, this is. That's all I know. Okay, so this is the result of the gel electrophoresis, okay? So I am gonna see what's going on here, okay? So first, I know that, okay, the three thing with the non-reducing one, they weigh 200 kilodalton because I only have one band. So I know that the whole thing weighs 200 kilodalton, right? But if I have like a multiple, then I have to add them up together, okay? All right, so this is a 200 kilodalton. And then I move on to the reducing one. I see that it's broken into different fragments, okay? And then it adds up, like 20 plus 180 is 200, right? So now, and then also I see that that's two fragments. So I can know that two of them are 180. And then another one is what? Is 20, right? And then I can also know that we have a disulfide bond here, right? Which is this, right? Which is this. So that when you run the SDS non-reducing, this bond cannot be broken. But when you run the SDS reducing, this bond is broken so that you have these two fragments, okay? So now we know the smallest subunit of this protein is what? It is 20 kilodalton, okay? So how many amino acids do you have in this subunit? Now we are gonna do the math, okay? So how much does one amino acid weigh? One single amino acid weighs 110 dalton, not kilodalton, right? But this subunit really weighs 20 kilodalton. That equals what? 20,000 dalton, okay? Then if you do the division, 20,000 divided 110, then you get 181, okay, roughly. So C is my best answer, okay? All right, I hope that makes sense. That's mellow, right? Okay, now this one is pretty heavy again, okay? But we've got this, okay? So this one reads, I have 4 proteins, okay? So protein A is a heterotetramer connected all by ionic interaction, okay? Each subunit weighs 10 kilodalton. And protein B is a homodimer connected by disulfide bond. Each unit weighs 30 kilodalton. Protein C is a monomer that weighs 25 kilodalton. Protein D is a homotrimer connected by disulfide bonds, okay? There's a typo here. This is bonds, okay? Each subunit weighs 20 kilodalton, okay? All right, so it's asking you to rank all proteins' speed of traveling in all 3 types of electrophoresis, okay? You can pause the video and give it a shot. I'm gonna take a sip of water before we dive into this, okay? Alrighty, so I think the best way to do this is to really draw out the 4 proteins, A, B, C, D, okay? So let's draw it out. So protein A is really what? It's a homotetramer. So 4 subunits, right? They're all the same, homo. Okay, so I have this, this, okay? And then I know it's connected by those ionic bonds, yeah, so which can be broken down by the non-reducing ones, okay? And then each unit weigh 10 kilodaltons. So this is 10, this is 10, this is 10, this is 10, okay? That's my protein A. What about protein B? Protein B is a homodimer connected by a disulfide bond. Okay, so it weighs 30. So this 30 kilodalton protein is connected by disulfide bond. And it's a homodimer, so 2 subunits, they're the same, right? Okay, what about C? Okay, so protein C is, it's a monomer, easy, okay? It's just a monomer, and it weighs 25 kilodalton, okay? All right, what about protein D? Okay, so protein D, it's how many subunits? There are 3 of them. They're all the same, okay? So I have a 1, 2, 3 subunits, and they're all connected by what? By disulfide bond, okay? So S, 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 S. Okay, so these are the 4 proteins, okay? So now let's draw it on the electrophoresis, okay? So first, the native one is really easy, okay? So protein A, B, and C, and D. So how much does protein A weigh? So each subunit is 10. You have 4 subunits, so it weighs 40, right? So you're gonna see the band here, okay? What about protein B? 2 subunits, each weighs 30, so it'll be 60, okay? So 60 for B, okay? What about C? C is easy, 25, right? So 25, it'll be here. And then what about D? D is also 60, right? Okay, so D is here. Okay, so that's the native one. Okay, now let's do the non-reducing one, okay? So the non-reducing one, let's do A, okay? Protein A, B, C, D, okay? So since we know that the intermolecular interaction between the subunits of protein A is really the ionic interactions. So this can be broken down by the non-reducing gel electrophoresis, right? Meaning that really those 4, 1, 2, 3, 4, all 4 bonds will be broken. After all the bonds are broken, each subunit weighs 10 kilodalton. So with the non-reducing gel electrophoresis, you will really see protein A here, right? Okay, what about B? All right, B is connected by disulfide bond. So non-reducing gel electrophoresis cannot break this bond. So it will still be what? 60, okay? Okay, what about protein C? Oh, that's easy, right? Super easy. I'll always just put 25, okay? And then what about D? Okay, D is also disulfide bonds connecting all the subunits, right? So there's no way the non-reducing gel can break the bonds. So it will be 60 as well, okay? Okay, now let's move on to the reducing one, okay? So the reducing one for A, you're gonna find A here at 10, right? Because the reducing one it can really break more bonds than the non-reducing, right? Okay, so that's A. And then what about B? Okay, B, this time the bond can be broken. Broken into what? 2 30 kilodalton subunits, right? So you have B here, okay? What about C? C is my favorite one, right? 25 here. Okay. What about D? D, this time, all the bonds will be broken. So 20, okay? Okay, 20. Okay, so to rank all the proteins' speed of traveling in all 3 types of electrophoresis, I'll say for the native one, C travels fastest, right? Because it's the lowest weight, right? So C travels faster than A, then B, and B equals to D, okay? And then for my non-reducing one, really A travels fastest, right? So A travels faster than next one is C, and then travels faster than B equals D, okay? And then for the reducing one, really, again, it's A that travels fastest, and then travel faster than D, and then C, and then B, okay? I hope it's helpful. It's a pretty long video. I hope you will learn something in this video. If you think it's helpful, please subscribe, like, and share. Thank you.